So my presentation goes a little bit off from what we were doing last week, which was a lot of integral sensory and survival analysis. But I love biology and methylation and genetics, so I chose something that was based on nonparametrics, but in a biology application. And so the title can be a little bit hard to get around, but detecting differentially methylated loci. So methylated, we have DNA that's methylated, and a locus is a location on the DNA, and we use that as coordinates. So every piece of the DNA is relatively mapped, and you can have a coordinate for that. And we want to test the difference between methylation at these sites among treatments. So multiple treatments, are they differentially methylated? And something that the title doesn't tell you is that they're helping to modify one of the nonparametric methods by accounting for age, because age is really relevant with methylation. So just a presentation overview. Okay, presentation overview. So first I'm going to go into what methylation is. So I'll first talk about the biology and the significance for statistics. And I'll also go into the data structure, because we need to understand the data that we're given in terms of methylation in order to structure our methods statistically and the research questions, both research questions and statistical questions. So then I'll go more into what the paper actually does. So we'll review existing methods and some of their flaws, and we'll adapt the Cusick test, which is the non-parametric test that is used now. And then we'll calculate overall p-values. So the contribution that they largely made was adapting this test and then showing a way to merge p-values in this setting. And then finally, we're going to assess performance by going through simulations and by going through ovarian methylation data. So simulations, they compare this to the existing methods and show that this method is better. And then they're going to use a widely used methylation data from ovarian cancer from the UK. So this is an open access data, and people use it very frequently when they're looking at methylation loci. So going into what methylation actually is. So if you think about it from a chemical standpoint, it's literally adding a methyl group, a CH3, to cytosines in the DNA. So we're not changing the DNA sequence at all. So we have A, T, C, G, and we're not changing that. What we're doing is we're actually just adding a CH3 to our cytosine. And so we don't change the sequence, but what we do change is the gene activity. So methylation tends to decrease the ability for a gene to be read, so we're kind of turning a gene off. And so by adding the methylation, we can control this gene regulation, and we can do that without altering the DNA sequence. Something that's important about methylation is the way that it's inherited. So you can't inherit it between generations. So a mother cannot transmit it to her daughter, father to son, parent to child. So you can't give it cross-generational. But methylation is really important because it's inheritable between tissue types. So a skin cell versus a brain cell, they're different. They have different functions. So they also have to have different proteins. And the methylation allows you to control the type of genes that are expressed in both types of cells. And so we want a skin cell to remain a skin cell, so the methylation is inherited between those cells, between those tissues. But again, it's not inheritable through parent to child. And also, if anybody has questions at any point, please interrupt me, because I like people to understand things as we go, or if there's like a review that you need, just let me know. So this little diagram just shows you, like, not really what it looks like, but what it kind of looks like. So you have these blue chromosomes, and if you super zoom in, you have this DNA, which is obviously red and blue in real life. And down here we have this methyl group, which is just amine. And if you have this methyl group on a gene, or on a C, it usually inhibits gene expression. So there's less gene activity that's around this methylation group. Whereas at this part of the DNA, there isn't a methyl group. So the gene activity is usually higher. You can have more expression up here. So that's the difference in terms of what methylation does. Again, not changing the sequence. You don't get it from parent to child, but it's very important to maintain the tissue cell type. Okay, so the biological significance. This is like one of the best parts. So something that's very unique is that methylation has a 98% association with age. And this was discovered by Dr. Steve Horvath, who has a joint appointment here in biostats and genetics. So what that means is as we age, our methylation changes, and you can predict which methylation loci are going to have an increase or decrease methylation based on your age. And that tells you like biological age, not just chronological age. So if a cell is old, you have an older biological age, and that's very well correlated to your own age. And so methylation is associated with age, and it's a very strong association, 80 or 98%. And so they consider age to be an effect modifier for methylation, because if you know the age, then you can predict some methylation. And age is also considered the strongest demographic risk factor for cancer. So we see most cancer is in older people because of aging. And cancer in young children, like that's not common. But you know, it still happens, but the idea is that it's not the most types of cancer. And so it's associated, cancer is associated with age, and so if we're looking at cancer and we're looking at methylation, we have to account for age, and that's what this paper is doing. And so methylation rules, some of the things that methylation is really important for is transcriptional regulation. So we all already mentioned how it changes gene expression. And it also has these other three rules. So chromosomal stability, which is really interesting. So if you have too much methylation or too little methylation, there are these hot spots in the DNA that fracture and then they break and they can translocate to another chromosome. So sometimes this is harmless, but there are very common genetic disorders where you have this cut in the DNA, so an unstable chromosome, that'll fuse to another one and it'll make a mutant protein. And so this is associated with a few types of cancers and a few types of genetic disorders. So if you have the wrong type of methylation, your chromosome might not be stable, and so that can lead to disorders in that way. Another way that it can lead to disorders is through almost all cancers. So cancer has to have all of these different abilities. It has to be able to move, it has to be able to proliferate, it has to be able to make vessels come to them so they can get nutrients. It has to have all of these abilities, but it usually starts by not being able to regulate their cell cycle anymore. And that we have specific proteins and specific genes that do that. So if they're misregulated at the methylation level, if something is not regulated, so we have P53. P53 is a cell death signal. If that is under-regulated, a cell won't kill itself, even if it's harmful. So methylation goes into controlling that. And it's very important for controlling other genes too. So whether or not you're dividing, whether or not you're growing, methylation is important for regulating all of the cell functions. So if one of those goes awry, it usually leads to cancer. And then the coolest thing is viral defense, because a lot of viruses actually integrate their genome into ours, and they will replicate by using our machinery and our proteins. And so once they replicate, they can go and infect other cells. But something that's really cool is that we can methylate the viral genome so that it's not expressed, so that we don't actually have the virus proliferating, and that's just a defense that has evolved within humans. And so methylation is important for so many different functions, so we have to understand the methylation in order to make progress in any type of biological aspect. And this image is beautiful, and it shows something really interesting. So this is from cancer. Every single chromosome has a fluorescent probe, and it's supposed to just be all one color, and they know what color the chromosome is supposed to be. So they took a cancer cell and they put these probes in, and what you notice is some of these chromosomes are actually fused together. So here you're missing chromosome 9, and it actually fused to 4, and then here you have another piece of 4 down here with 22. And so this is very common in cancers, which they're pointed out here and here, where you can kind of see this blue. I don't know if that's too dark on the screen. But this is just an example of what it would look like for chromosomal instability with regard to methylation not being regulated properly. That was a lot for biology, but basically it's really cool and it's important to understand. Okay, so now we understand why methylation is important. So now we have to talk about the actual data that we're getting. So methylation data are, the data that you get are fluorescent intensities for each locus. So each coordinate on the DNA gets these two values, M and U. M is the average signal from a methylated locus, and U is the average signal from an unmethylated locus. So they can range from 0 to 100 typically. If they're above that, then that makes the beta value not, it makes the beta value outside its bound, and so you usually view those as bad reads. But we tend to have M and U between 0 and 100. And beta is what we're interested in. Beta is the methylation level at a locus. And what you're doing is you're taking the maximum of the methylated value at the locus, and you're dividing it by, or you're taking a ratio with the maximum of the unmethylated plus 100. So you can think of the unmethylated signal as noise, and then you can think of this as the actual signal. So if you have no background noise, it's just the percentage of methylation. And so that's how they roughly interpret it in a biological atmosphere, is that we view the beta value as the percentage of a site that is methylated. And so that usually ranges, or it does range from 0 to 1. 0 being completely unmethylated, so you have no signal. 1 being completely methylated, where you have no background noise, basically. Yeah?
Just curious, so now in experiments, people can really record the fluorescent intensity from one locus?
Mm-hmm.
Or how do they do that?
Yeah.
On a microscope?
No, not so. That's a good question. It's very interesting. So you have ACTGs, and it's actually called bisulfite sequencing. So what you actually are doing is the cytosines that have a methyl group on them, when you apply this bisulfite treatment, they will not react. And so the Cs with the methyl on them will stay Cs, but the Cs without methylation on them will convert to Us, or convert to another base pair. So that then, after you do this treatment, you'll run it through a DNA sequence, and the DNA sequencer uses fluorescent intensities in order to detect which nucleotide is there, and the ones that were not converted will read as Cs. And so all of the Cs in your sequence end up being red, and you can tell that those fluorescent intensities for the Cs are actually the methylated ones.
Cool.
Yeah, it's really interesting. Yeah, and it's pretty precise. So you can get the data, and you can see the fluorescent intensities, and if they start to get wiggly, you can tell, okay, this probably needs to be red again, because there's a lot of noise that's going on. But typically, the signals are very well amplified. So it's very precise, and they've done a really good job at improving this method. Yeah, absolutely. Yes?
So this is a percentage between 0 and 1.
Mm-hmm.
Can it actually be 0 or 1? Can, right?
It can, but it's pretty rare because we tend to have a little bit of background noise.
Okay.
Yeah.
So you won't be like 0 inflated or 1 inflated.
Correct. So they tend to hover around 0 and 1. I think that might be on the next slide. Yeah, so they tend to actually cluster close to 0 or 1. So these locations on the DNA tend to be either completely unmethylated or completely methylated. And so they tend to be clustered like this. And other data, sometimes it's around like 25% or 50%. But we know that this is how the beta values usually are structured. And so because we know this changes the type of test that we actually have to use. So looking at this graph and knowing that the beta values tend to cluster around these values, we can immediately say, okay, this is not a normal, beta is not normally distributed. If you have a lower bound, then you have to be, well, you're very likely to have a skew. You have an upper bound, you're very likely to have a skew. And then also, you can't be normal because it's bounded. So normal has these infinite tails, and so clearly you can't, like normal is not a good approximation. Which means that the common methods that they use that assume a normal distribution have low power. So ANOVA, linear regression, and t-tests, they have low power to detect these differentially methylated loci. And again, just to reiterate, we know methylation is very strongly correlated with age, so we need to take that into account. Okay, so the purpose of this paper. We know DNA methylation changes disease development, and we want to understand that. But the current statistical methods don't allow for one of these things. So you might allow for trend testing, account for aging, or you might be nonparametric, but you don't allow grouping. Or you might allow grouping and aging, but you don't allow trend testing. So the statistical methods are just missing some key component, and this paper addresses all of these. And so the goal is to develop or modify a nonparametric trend test to detect differentially methylated loci under multiple conditions that account for age. So we're making a test that doesn't assume a distribution on beta. That's the nonparametric part. We're looking for a trend across treatments. So the idea is that we have like placebo, and then like dose 1, dose 2. So the idea is that we're looking for a trend in methylation. So is methylation lower? Here's beta. So is beta lower here? And is there a trend like this? That's what we're looking for. So nonparametric trend test to detect this difference across treatments. But we're also going to account for age. So what we're doing is we're actually going to be grouping these into different age categories and then merging them back together because we want to account for this important covariate in the model. So that's their goal. Does that make sense how we have both treatments and age in this model? Okay. And again, if I need to go over something, no worries. So the contribution that they made specifically, they're showing a method that's good for non-normality of beta. They're adding age as a covariate, and they're looking for a trend of beta, which is the methylation percent across these different treatments. And the way that they do that specifically is modifying this nonparametric trend test with an age covariate, and they provide more power in detecting these loci and also decrease the variability because they're adding age, which is a very good way to account for variation in beta. And so that decreases the variability in your estimates because you're removing some of that, you're putting some of that error into a covariate. Yeah?
So it's bimodal thing there. So that is the idea, they account that by using age, is the idea?
So what actually ends up happening, so this is like over many loci. So each locus usually has either this mode or this mode. And this is just many different spots overlaid. It's hard to see all the different colors. So sometimes they're bimodal, but they're typically just one and have a skew. Okay, so the limitations, as we already kind of mentioned, we can do a Kruskal-Wallis. So if you're thinking about, okay, it's not normal, so let's find something else. And Kruskal-Wallis is good for ranking things, but you can't determine the difference between things. You can just say these groups are different because of the ranks. ANOVA, again, assumes normality, and so if we don't want to put that assumption on it, we need to have a different method. And then linear regression is very commonly used, whether it's GLM or regular linear regression, but age is usually put in as a continuous variable, and that actually performs poorly. And it's not really known why it performs poorly, but it doesn't perform well. And this is a graph from a different paper. The way that they say performs poorly is whether or not your significance is the same with two different types of methods analyzing the data. And so there's this 45-degree angle, or line, and you expect the values to be on that, but you can see it's like a 30-degree angle between a t-test and regression with age. And so these don't give you the same results, and they need to improve the method somehow. Okay, so the Kuzik test was developed in 1985. It's a Wilcoxon type test for trend, and it accounts for having trend in 3 or more independent groups. So this would already allow you to do placebo, dose, and dose 2. The Kuzik test is nonparametric, so it doesn't assume a distribution of the variable. It leaves it unspecified. And in our case, we're interested in beta, so I have beta in the parentheses. It is considered the locally most powerful test for location shifts under the logistic distribution. And we like the logistic distribution because beta is bounded from 0 and 1. And then the test statistic itself is a weighted sum of the ranks, so the Wilcoxon piece, and it's weighted by the group's score value. So if you can assign the trend value 1, 2, 3, that's the weight that you're given to the ranks because you expect them to be going in some order. in order to use this test, you have to have, there's assumptions, of course. So your data must at least be ordinal. So you have to have a numeric value that you can rank with. And that's important for the ranking piece. Your groups that you put them in have to have a meaningful order. And you have to specify that a priori. And the way that you can score them is, you can do many different ways. So the typical one is just doing 1, 2, 3 up to G. But you can also do it based on effect size. So if you think placebo has like a 0.1 effect size, you can do 0.1, and then if you think dose 1 is going to have like a 0.5 effect size, 0.5, dose 2, 0.8. So they don't have to be uniformly spaced. And it gives you a lot of wiggle room in terms of finding this trend. Something that I couldn't find is whether or not your variance has to be equal. So I kept searching. I couldn't find if your variance across treatments had to be equal. But one way that they address this is by using the beta distribution, which allows for heteroscedasticity, but then accounts for it. So they show that it's robust even to a little bit of variation in the variance. The last assumption is that your errors have a logistic distribution, which is just important for this type of test so that we can range it from 0 to 1. Okay, so the hypotheses based on this test. Your null is that a locus is not differentially methylated across treatment groups. And this is for the basic test that they aren't adjusting yet. And so that means you're assuming that under the null, the beta values at each of these treatments are the same. And then the alternative is that there's a trend. So like we had already said, either beta 1 is less than beta 2, less than beta k, or the opposite. And so you're testing equal versus one of these directions. So this is actually a one-sided test. The modified test that they're presenting, the only difference in these hypotheses is that you're controlling for age. So not differentially methylated across treatments after controlling for age. There is a trend, they're different after controlling for age. So that's where this is going. So the modified test, the way that they go about this is they first group people into these age categories, and they chose 6 age categories. They chose this after the fact, but they chose it between age 70 to 75, etc. So they chose that before looking at the data, but they just have these age categories. Then they're applying this modified test, and you're applying this modified test, which I'll show you. And you get a one-sided p-value because we're looking for a one-directional change. And the direction is expected to be the same. So we're testing for a monoclonic trend, so we don't expect something like this. We only are looking for things that are like this, and they don't have to be linear either. They just have to be monoclonic. And then using this modified test that groups into age, we're going to calculate an overall p-value, and then we're going to compare to current methods using simulated data and then the real methylation data, which we mentioned from the UK ovarian cancer data. Okay, some terminology. So we've already started talking about K and G, but it's important to go through all the other variables you're about to see. So K is going to be the treatments, so placebo, dose, etc. G is going to be the number of age groups. In this case, we have 6. And then N subscript G is the total sample size in the age group G. And then that can be generalized to just big N being just the total sample size, which we're used to see. And then little n GK is the sample size in age group G, treatment K. So, we're narrowing this down. If you think about it as a matrix, it's, if we have 6 age groups in 3 treatment types, it's a 6 by 3 matrix. And we're saying NGK is one of those cells. RGI is the rank of the ith subject in group G. And something that's important to recognize here is if we have 60 subjects, the ranks do not go 1 to 60, they go 1 to NGK, or NG, sorry. So it's a rank within a group. So it's not 1 to 60. If you have 10 people in a group treatment, it goes 1 to 10. SGK is the score of the treatment type. So 1, 2, 3, or that could be the effect size, 0.1, 0.5, 0.8, and that's the score. And then finally, we have this weighting factor, because you want to take into account how much contribution each group and treatment is having. So you just have this weighting factor, which is the number of people in that group, in that treatment, for that age group. Yeah?
So for each age group, you have different scores, right?
Yes. Yep. So if we so then we're going to have age here, and then we can have, I don't remember what ages they use, but say it's like 15 to 20, 20 to 25, blah, 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 blah. So then we're going to have, in this entire age group, we're going to end up going through all these different treatments. So if you have 10 subjects here in placebo, then you're going to have ranks, and you're going to, all the ranks are going to be multiplied by the score. And so let's say the score, this is 1, 2, 3. So we're going to have the ranks times 1 in here, and then we're going to add that to the ranks times 2, ranks times 3. And so this is, you can already see how this is about to go, so that if you have an increasing trend where you expect the beta to get larger as you increase in these treatment types, that means you expect this value overall to be bigger, because you're going to have more of the larger weights, or the larger ranks over here. Whereas if you have a decreasing score, that means you expect the smaller ranks to end up over here. So you're going to have a smaller overall score compared to your null value of like equal betas.
Yeah, so and this score has to be pre-specified.
Yes, absolutely.
So if you have many age groups, between different age groups, is there a rule of thumb how you pick the scores?
So the scores will be the same across all age groups.
Across all age groups?
Yeah, so if there are, say there's 8 subjects here, you still multiply their ranks in this age group, so ranks of group HG times this treatment number. Yeah, 1, exactly. So
I saw the general notation, you actually allow a G there, so that's why I was wondering.
Oh, in RGI?
Yes. SGK.
Oh, SGK. Yes. So in the GF age group, the score that is associated with it. So notation switches back and forth between SGI and SGK. The idea is that SGI is when you're calculating your value, and then SGK is used in the null situation where it's like, okay, we don't have any difference across treatments. And so that can get a little confusing, but that was the way the paper did it, so I was trying to stick with some of their notation. Okay, so that's notation, or terminology, and then I'll actually now show you what they're doing. So recall it's a Wilcoxon type test, and it's testing for trend, monotonic trend in beta, over these different treatments, and we're looking at it from a one-sided direction. So the actual value for this age group is summing over all of the people in that age group, and it's the rank of that person in the age group multiplied by the score that they're in. So 1, 2, or 3 for the score, the rank of that person in this age group. And then the overall value, you can see that this kind of looks like a CLT, which we'll get to soon. But you have this T of G here, which is the value for that age group, minus the expected value. And the expected value, you can see there's this Wilcoxon part here, so the ranks. Wilcoxon is the 1/2 n times n plus 1. And since we're doing it for groups, it's just n little g. And then this part is the expected value of the score. And so if beta is the same across all of these treatments, we expect the proportion of people in that age category, in that treatment, to be multiplied by the score. So the proportion of people in dose 2, age group this, is going to be multiplied by the score. And that's what we expected with the null. So we subtract the expected value, and then the variance down here. So we again can see the Wilcoxon piece here. and then we're multiplying by the variance of the score, so S, which is just by definition. So E of S squared, or like the expected value of the score, so S squared W squared minus expected value squared. So they're just using the actual definition down here. Okay, so I wanted to show you a comparison between the original and the modified. So original is a test statistic for one methylation locus, and it's just if you have locus 1 at DNA coordinate, I don't know, like 300 something, blah, 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 then that's what this is. And then the modified acoustic test, I'll go a little bit further through because that's what we're looking at in this paper. So this is for one locus and one age group. So T sub G. So we're getting a T for one of these rows, and we're going to get 6 of them because we have 6 age groups. So again, TG, summation, the ranks times the score in an age group. Under the null, we expect that to be this Wilcoxon piece that we just mentioned in the expected value of your score. And then the variance is, again, the Wilcoxon piece times the variance of S, which we saw on the other pages just by definition. Okay, and so we saw the CLT in there, so this makes sense. But if you do T of G minus the expected value divided by the standard deviation, you expect that asymptotically to be about normally distributed. And so that means we can get a one-sided p-value by just using the CDF of a normal. And so the probability of this test statistic being larger than Z, you use the CDF of a normal. So that makes sense. Now let me see it. Okay, CLT. So we're getting a p-value by just using the normal distribution. But the problem is we have 6 p-values at this point because we did this for each age group. And so what we're interested in is not whether or not they're differentially methylated across age groups. It's, are they differentially methylated across treatments? And so grouping them into the age was just to allow us to adjust for this. So now we have to merge all of these age group p-values down into one p-value that detects whether or not they were different across the betas. So K treatments, G age groups. Right now, G p-values, number of statistics for each methylation locus. And something that we have to keep in mind is that the p-values for each age group are independent. So we started with an independent sample. And so just by grouping people into the age categories means that they're still independent. So that means we can combine the p-values by just multiplying them. And the way that we're going to do this is by Fisher's method. Fisher's method is widely used. It's the idea that if you take a uniform variable, which p-values are normal, and you take the log and you multiply it by -2, that's chi-squared. And I'll show you the proof of that soon. So what we're going to do is for each age group, so we have 6 of them, we're just going to multiply the p-values, which also is summing the log of the p-values. And that's going to be chi-squared. And we're going to use this distribution to get our p-value overall. We're just going to use the chi-squared distribution, which means we just changed G dimensions to 1 dimension so that we can test the significance at this specific locus. So into the proof that if you take -2 log the product of your p-values, that's chi-squared distributed. So if we have y is equal to negative log x, where again x is uniform, then that means you can take the inverse, and the g inverse of y is e to the negative y. And then we want to take the derivative, so that's negative e to the negative y. And from this formula that we're very familiar with from 202a, you can get the PDF of y by f of x, taking the inverse of it, and then multiplying by the Jacobian. And since x is uniform, that means f of x is just 1. And then the Jacobian is just the derivative. And so the absolute value, you're left with e to the negative y. And we recognize that as exponential with a rate factor of 1. And so that's good. But what we know is that exponential 1/2 with the rate 1/2 is chi-squared. So all you have to do is add this -2, and then you'll end up getting e to the negative y over 2, which is your chi-squared that you wanted. So that's why we have the 2 in front. We get chi-squared, which means that overall we can just get our p-value by -2 summation log of p. It's pretty nice. Okay, so just taking a step back, to calculate the overall p-value, we first knew that CG, one of our test statistics for each of the age group, is normally distributed. So we did a one-sided test for Chen to get a p-value using the z. Then we're merging them. But what we have to take into account is the trend direction. So if we're doing a one-sided test, recall we said if you're increasing, you expect C of G to be larger than the null, which means you expect your Z to be on the right side. Whereas if you have a decreasing trend, then you expect C of G to be small relative to the null. So you want a left side p-value. But in practice, we don't know what direction that's going to be. So we'll get to that. But the idea is that PRG, you want the right-hand probability. And then for PLG, you want the left-hand probability. So the idea is you want either one of these p-values, but you don't know which one you want. So the way we account for this is we say, okay, so we know that the direction matters. So W is just a new test statistic that we're going to be looking at. -2 log of product 1 minus PL of G. So if you're increasing, you expect this to be large, and so you want this right-hand probability, so 1 minus. And then W decreasing, you want the product of the left-hand probabilities. And the idea is that we can just use both of these and choose the maximum, but they're obviously associated with one another because both of these actually are using the left-hand probability instead of this one using the right-hand side so that we can stay consistent. And so that's why we're subtracting left-hand. And so we're using both of these. They're chi-squared. They're not independent, but we'll get there. And so what we're doing is we're just going to choose the max, which is not conservative. And the reason it's not conservative is because they're trying to detect loci. They don't want to be conservative here. They want to detect even small variations in methylation. So they're taking the max, which is not conservative. And then the only conservative piece here is that in the end what they're doing is they're choosing the upper bound for their overall p-value. And so I'll show you how we get there. I feel like this is helpful, so I'm going to do it over here actually. Okay, so we want the probability of W, and we're arbitrarily choosing, well not arbitrarily, sorry, we're choosing the max of either the increasing or the decreasing value. And so if we look at the probability of this W being greater than W, W is a max, so what we're doing is we're going to find a lower bound for the max, because here we want to be a little bit conservative. So the probability that your test statistic is greater than this is the probability that W1 is greater than W, or W2 is greater than W. And just using typical like set, I think it's set theory, I don't know, but we know that you can get this probability by taking the probability of W1 greater than W plus probability W2 greater than W, and then minus the intersection. And we know that this is true. But now we want to find a bound. So this is always going to be, if we don't take the intersection, that means it's going to be less than or equal to the probability of these two. So less than or equal to a probability, I'll just, for shorthand, probability P1, or W1, W2. And both of these are evaluated at little w. So we're doing greater than, which means we have to do 1 minus chi-squared 2g evaluated at w, and then plus 1 minus chi-squared 2g evaluated at w, and that's just 2 times 1 minus chi-squared 2g w. So this is a lower bound for the max, which is what we're using. And that's how they're getting their overall p-value for each locus. We can thank Joseph for helping me on that. Okay, so now we're going to move into how they actually assessed this method. So we're going to go into their simulation. So they simulated data. They used 3 different treatments because the ovarian cancer also had 3 different treatments and 6 different age groups. So they're going to simulate with 3 different treatments, 6 different age groups. They're going to just score them 1, 2, 3. And what they're going to be doing is, you'll see this later, is that they reverse and like change the score order and show you that it performs poorly if you don't know the score ahead of time. But so we have treatment scores 1, 2, 3, and they're going to have 3 different distributions that they are simulating beta values from. And so in each of these simulations, they either have a uniform, truncated normal, or beta distribution, but you allow them to have different parameters so that they can have a trend. So you're simulating treatment 1, treatment 2, treatment 3. Either they all have uniform, but they have different values so that you can detect trend. So uniform is good because it's flat and just it's one way to show that this is good. Truncated normal, so if you want to use normal, I don't know why they ended up using this because we don't really like the normal distribution, but it's truncated, so it's allowed to have a 0 and 1 bound. And then the beta distribution is the best one because that allows heteroscedasticity and also allows the bound naturally. So those are the 3 different ways that they're going to simulate the data, 3 different distributions. And they use small sample sizes because that's typical. So when you get methylation data, you have thousands, hundreds of thousands of methylation loci, but not very many people. So they used small sample sizes, and they showed that it works both in equal sample sizes and unequal among treatments, which we don't really expect equal, so it's good that they looked at unequal. And then they compare this to how you would do it under ANOVA and Kruskal-Wallis. Okay, so again, I just like to reiterate what we're doing so that it's clear where we're going. So we're detecting a monotonic trend in beta over these 3 treatments. So they're going to simulate beta values with either an increasing or decreasing mean value. And so I have an example here that they're going to take 20 beta values from a normal 0, 1, 0, 1, 0.25, 1 for each of the treatment groups. And then they're going to do all of these at an alpha equals 0.05 with power. Oh wait, no, they don't do power, they just do empirical power. And they do this 10 to the 4th times, so simulate a lot so that they can get a relatively good number. So, can you see this? Yes? Cool, okay, good. Okay, so this is the results using empirical type 1 error. So, what they did, if we look at the uniform UAB, so they're talking, left endpoint, right endpoint. And so they have these different values, sample size 20, 20, 20, so equal sample size. You can see that they all do pretty well at being at the 0.05 level. So of the times that the null was true, how many times did you reject? Like the percentage of times that you rejected. So that's the empirical type 1 error. And you can see it does a pretty good job. It's in between ANOVA and Kruskal-Wallis in terms of performance because ANOVA tends to hit pretty well at 0.05. Kruskal-Wallis tends to be an underestimate, and then the method tends to be in between. So like 0.51, 0.051, 0.045, 0.047. So you can see it's kind of in the middle for this. And it's the same thing across the 2 different types of distributions that you have. So it's the same for the truncated normal, performs well, and for your beta. So empirical type 1 error is good. Now we're going to go to empirical power, which is what we really want. So when the alternative is true, how many times did we get it right? So it's sad. ANOVA and Kruskal-Wallis have really low power. And right now, let's just focus on this first column of new, which is their method of the modified Cusick test. You can see it has pretty, it does better. Sometimes it does pretty poorly, 0.6 power, but overall it does a lot better than ANOVA and Kruskal-Wallis, which like 0.48, 0.38, 0.36, 0.62, it's not very good. These 2 methods are not very good at finding these differentially methylated loci. And again, It's across equal sample sizes, unequal, and 3 different types of distributions. And so I asked you to focus on this first column because that's where they correctly gave the scores. So they gave them 1, 2, 3. It's pretty small here, but they gave the correct scores to the treatments. And what they show is that if you mess it up, it's pretty bad. So 1 to 3, 2, when 3 is supposed to be either the largest or the smallest, it performs really poorly. So the only time that this test is good is when there actually exists a trend. And if there is a trend, it's really good. But these are not good at identifying trends. This new method, modified Cusick, is really good at identifying trends if they exist. So that's, you know, a little like, oh, do we really think that there's a trend? I don't know. But
so 3 is the one that messed up.
Yep. So this one was still fine because they just did 1, 1, 2. So they said the first 2 are equivalent, and then just the last group is different. So,
yeah. So currently, this is what is supposed to, what you expect to see, right? So when you design something specific for a particular type of trend, when you do have that trend, you have more power.
Exactly.
While the ANOVA and the cross-covalence is just detecting a difference.
Exactly.
So it's the alternative is more general. So it's quite stable in terms of the power, but when there's a trend, it won't be as powerful as the one that you.
Right. That's a good way to say it, that it's very stable for detecting the difference, regardless of trend. But exactly, the test that they're making is for trend specifically and for difference. So exactly, it's more powerful because we expect it to be. The other one, like you said, is more stable, which is a good way to put it. So that's the empirical power, and this was simulated data. So now if we go to real data, we're going to use UK's ovarian cancer population study. It's DNA methylation profiles across all of these CpG. So that just means the sites of methylation. So typically we call them CpG sites because more biology, it's fun. CpG, G bonds to C, C to G. And so if this one is methylated, then that means this one gets methylated. So it's just a way to methylate both sequences, so it's called a CpG, because you want the C's on both sides of the DNA to get methylated. So that's why it's CpG. I didn't realize I kept that notation in, so my apologies. And they're looking at postmenopausal women with ovarian cancer, and they match them to healthy controls. And the way that they're grouping these people is of these women that have or had ovarian cancer, did they receive treatment, or are they in the stage where they haven't received treatment? So pre-treatment, post-treatment, healthy age-matched controls. That's what they're looking at. And their age-dependent DNA methylation is, they know that it's suppressed in stem cells, and that's a hallmark of cancer. So like we talked about, if you have poor methylation regulation, that can lead to cancer. And they knew this ahead of time, so they wanted to account for age. And so pre-post control, yeah, I can get rid of that. So pre-post control, it first is not intuitive the way that they're ordering them. So the way that they're ordering them is pre-treatment, post-treatment, and then control. So when you first think about it, or the way that I first thought about it, is I expected it to be control pre-post. But the reason that it's scored 1, 2, 3 is that if a woman has ovarian cancer and they haven't been treated yet, you expect them to have the worst type of methylation control. So you expect them to be like either have way too much methylation or way too little. And then people who already received treatment, that means they're closer to being cured, or they are cured. So you expect them to have an intermediate level where they have better methylation control. But somebody who's never had cancer, you expect them to just have good methylation control. So that's why it's ranked in this order. So pre, post control. And so I just wrote worst, better, best methylation. I don't say less, bigger, more, because again, it's two-directional. It can either be greater or less. Okay, so they noticed, or well, they expected this, but they detected more loci with their adapted method. So when they say T1, T2, T3, that's just the test that they used. And then what they're doing is they're saying, okay, using the Fisher's method, p-value, and they have different cutoffs because you're detecting 27,000 methylation sites, so you have to control multiple testing somehow. But they said, okay, it depends on how, what your criteria are. So they made them into different components based on how much you wanted to see. And so you can see that the ANOVA, 981, Kruskal-Wallis, 1300, and then the new method, 2,900. So it detected a lot more. And then what they're doing here, T1 and T2, is the number of sites that were identified the same. So between ANOVA and Kruskal-Wallis, 926 sites were the same. Between ANOVA and the new method, 931. Between Kruskal-Wallis and the new method, 1,294. So you can see that not only does it detect more, but it detects the same ones that the other test did. So that's what we want, obviously. And then overall, all of them, 895 if you compare all of them together, which is pretty good. But they showed, okay, so it detects more, and it detects the same ones. So we should use this one. That's what they showed there. And then this is a plot, so 6 different age groups. In the end, you're testing whether or not they're different across, if it's different across treatments. So they merged them all together, but they wanted to show how they were doing this. So this is mean beta value. And so pre-post control. You can see across all of these age groups, it's changing this direction. And so there's a trend in all of the age groups, but the value that it starts at is slightly different, which may be important for detecting the difference because you're not controlling for age in the other methods. So that's why they have them boxed out like this. Different age groups, you see a trend is consistent across all of them. And they're only plotting the ones with, they have like a low cutoff of 10 to the minus 3. So they're being, a little, they're not being conservative in any of these methods. So they're plotting the least conservative way. So, yeah. But you can see that there's clearly a trend, and this test did a really good job of distinguishing that. And reproducibility is obviously key. And so they provided their code, and the only things you have to input are the data, so your methylation data, the number of age groups, so how you're going to dichotomize age, the scores for your treatment groups, so 1, 2, 3, 0.1, 0.5, 0.8, and then number of subjects per age group, which you can also just tell it to read that in the data step, but they chose to just say, okay, number of subjects per age group, and then it outputs a group p-value, so the overall p-value, or the group p-value and the overall p-value. So the p-value for each age group, and then it gives you the overall p-value after it condenses it. And the ovarian cancer data is a nice source because it's open access, so you can just reproduce their results. And they tell you exactly what they did. They removed 60 subjects with bisulfite values less than 4,000 or coverage less than 95%. What that means is if you had a bad methylation read, that's bisulfite values less than 4,000, or if your DNA for some reason, we didn't get a very good read on it, maybe your DNA was just really, really bad in terms of cancer, and there's a lot of translocations, and we just couldn't cover very much of it. So they're looking at only data that were relatively robust and relatively well maintained. Okay, so just a simple recap of benefits. This is more powerful if a trend exists across these treatments. It outperforms existing methods, again, under the caveat that there's a trend. It adjusts for age, which is important for methylation. It's nonparametric, so your beta can take on any values. And this allows you to detect loci with small effects. So The other methods could only detect like 800 loci. This one could detect 1,300. So you can even detect small differences in methylation, which might have important implications in the disease development. And this is important because a lot of biological like logic plans take methylation as an input. So if you say this loci has 85% methylation, then we can expect this much protein to be output. And the idea is that they're trying to make these cell signal pathways. And so if you have methylation as an input, even if it's relatively small, that allows you to better predict what's going to happen. Okay, so just the conclusion. Paper gives a modified statistical approach for detecting methylated loci with multiple groups. It's based on this Kuzik test, which is robust if a trend exists. And they showed that this performed well with simulated and real data. And it outperformed both Kruskal-Wallis and ANOVA. And that is it. Questions?
How can they put other factors influence the DNA's performance expression? For example, DNA acceleration?
How do they include other effects of DNA regulation, you're saying?
Yeah.
So they aren't. So the idea behind this is they're only looking at the methylation. So you're absolutely correct. If you're looking at the amount of protein that's output, you would have to take into account like transcription factors and other things. But they're actually just looking at methylation because methylation has the strongest control over gene expression, whereas transcription factors, they're important, but methylation is the strongest. So they don't consider any of those other things. But if you were going to a different scenario where the biologists are doing these set-based analyses where you put methylation in as a predictor, then you'd also want to put in what you're suggesting with these other transcriptional regulations. Yeah?
So just wondering, in Dr. Horvath's molecular club work, it's not that 98% correlation between methylation and age is not real age.
It's biological age, correct. But biological age and chronological age are also very well associated. So it's hard to actually use biological age because it's hard to measure that, so they use chronological age.
Oh, okay.
So the idea is that they want to explain the biological age based on methylation, but we only know chronological age.
Yes. And just in his original work, how did Dr. Horvath measure biological age?
So he used linear regression. And I feel so bad because I'm like, linear regression sucks. But his work,
confused.
yeah, so it was just basic linear regression. And the reason he didn't improve it is because it was 98%. Like, why improve it?
So that linear regression is between your chronological age and the methylation.
Yeah,
It's
methylation
already
sex.
98%.
Yes, yeah. So his work wasn't detecting for a trend. It was only looking at, okay, how is methylation associated with age, with chronological age? Yeah. So age in that case was continuous, but they were also looking at a slightly different question. But now they're looking for a lot of differential methylation. Anything else?
And so here you have thousands of loci, right? And use a multiple comparison idea to control for the lower background error. And you want to be, in this case, aggressive.
Yeah.
Yeah, try to do that. And so this type of approach is more, you do locus by locus, right? Each one, each location you do a test.
Yes.
Test, yeah. And so it's more a marginal approach. And so between different loci, biologically, I have no idea here. Are they considered independent or they could be dependent?
So it depends. So you can think of it like in terms of GIS data, like things that are nearby are more dependent. So usually these, of course I erased it, the CPG sites, it's usually like a CPG island. So it's just CPG, CPG, CPG for like 20, 30 base pairs. And so usually that entire island is either methylated or not. So nearby, it's dependent. but as you move away from a site in a gene, they're independent. And most of these the methylation sites that we look at are usually spread out. So the ones that the loci that we're looking at are considered independent.
So actually it's like
Yep, so they're pretty far apart, so you can say that they're relatively independent. Or a term that we recently learned was asymptotically independent. Okay, thanks guys.
All right, thank you. Well, excellent. So, I enjoyed this more than I had my own lecture.
Biology's cool.